Hello and welcome back. This is our second video in our video series on recombinant DNA technology. Let us continue with 8.2, methods in gene cloning. So 8.2 is a continuation from 8.1. In 8.1, we learn about the tools in recombinant DNA technology, and now we're continuing with how to use the tools. The learning outcomes for this subtopic is that I hope you would be able to give an overview using a diagram to show the steps in gene cloning by using plasmid. And I also hope that you would be able to describe each of these steps in gene cloning by using plasmid as the vector, using these 6 following steps. Okay, so first things first, before we learn about the details of gene cloning, what is gene cloning? So you know the word cloning, right? So perkataan clone means kita ada sesuatu dan kita mau menggandakan sesuatu itu untuk membanyakkan dia. So to clone is to make copies. And gene cloning is more or less the same thing. It is the production of multiple copies of a single gene. Kita ada satu gene, kita mau perbanyakkan untuk dapatkan banyak salinan. There are 2 purposes why we want to do gene cloning. The first is so that we can make many copies of that particular gene, and the second is to produce a protein product from it. Okay, 2 sebab kita mau kasih banyak gene, dan bila ada banyak gene, kita boleh hasilkan protein daripada gene tersebut. And this is usually expressed in this kind of diagram. We've been seeing this diagram since 8.1, and we're going to use the same tools that we learned about in 8.1 to do gene cloning. Jadi ini adalah recombinant DNA technology yang kita belajar dalam 8.1. Benda yang sama, coba sekarang kita belajar dalam setiap step dia. So as I told you before, there are 6 steps for gene cloning, and we are going to look at them one by one. The first step of gene cloning is isolation. Isolation means mengeluarkan, mengasingkan. We are going to do the isolation of plasmid and target DNA. Plasmid yang kita gunakan adalah plasmid yang kita belajar dalam 8.1, yaitu PUC18. Dan target DNA yang kita akan gunakan adalah insulin. So we're going to use human insulin as our target DNA. So once we isolate them, we can go on to the second step. The second step is to cut. We will cleave or cut the plasmid and the target DNA with the same restriction enzymes. In this one, we're going to use the enzyme EcoR1. The third step is insertion. We're going to insert the target DNA into the plasmid to form recombinant DNA plasmid. And then, of course, we're going to stick them with DNA ligase and then continue with step number 4, which is transformation. Transformation of recombinant DNA into bacterial host cell. So kita masukkan recombinant DNA plasmid itu ke dalam host cell yang kita mau gunakan. Host cell yang dalam contoh ini adalah E. coli. We're going to continue with the 5th step, which is amplification. Amplification of recombinant DNA in the bacterial host cell. So dalam bacterial host cell itu, plasmid akan berganda. Bila dia diperbanyakkan, itu adalah amplifikasi. When it increases in number, that is amplification. And last but not least, it is screening. Screening is choosing which ones are the ones we want. So during screening, we will plate the bacteria on agar plate containing ampicillin and X-gall, and we will do blue-white screening based on LEC-Z selection to obtain our desired cloned cell. But don't worry if you're confused now. We're going to learn about each of these steps in detail after this. Okay, so here are the 6 steps, and you can remember them more easily by remembering ICITAS. ICITAS ka, isitas ka, macam mana kamu mau ingat? Ingat lang, ICITAS is for isolation, cut or cleave, insertion, transformation, amplification, and screening. So this is a continuation from the diagram just now. In the diagram just now, it doesn't show where DNA ligase is being used. So this additional purple and orange diagram is to illustrate where we use the restriction enzymes and where we use the modifying enzyme, in this case, DNA ligase. So, ini adalah diagram tambahan saja sebab diagram sebelah kiri itu tiada tunjukkan di mana kita gunakan DNA ligase. Ini untuk memberikan gambaran sahaja. Alright, so let us continue by talking about each of the steps in gene cloning. We're starting with the first one. Kalau ada orang minta kamu describe steps in gene cloning, describe isolation, you just say, isolation is isolation of target DNA and cloning vector or plasmid where two different sources are selected. First source is your target DNA or your gene of interest, isolated from donor cells. Donor cell can be human, animal, or plant tissue cells. So, kita keluarkan target gene yang kita mau daripada sel manusia ataupun sel haiwan ataupun sel tumbuhan. And second, we will isolate the cloning vector, or in this case a plasmid, from the bacteria. So this one is plasmid PUC18 isolated from E. coli cell. Remember in 8.1 when we drew that plasmid, okay, saya minta kamu lukis dia punya MCS, lukis dia punya LEC-Z, dan lukis dia punya AMPRG. Well, now we're going to go back to this and we're going to point out that the cloning vectors or plasmid carries two useful genes that are going to be important in the steps later on. Dua gene yang saya minta kamu lukis itu adalah AMPR or ampicillin resistance gene, which codes for resistance to the antibiotic ampicillin. Asalkan bakteria itu ada AMPR gene, kalau kita letakkan bakteria itu dalam medium yang mengandungi antibiotik ampicillin, bakteria yang ada AMPR gene boleh hidup. Kalau kamu ambil bakteria yang tiada AMPR gene, kamu letakkan dalam tempat yang ada antibiotik ampicillin, dia akan mati. So basically, this gene gives it the ability to survive in antibiotic. The second useful gene, the green color one that I asked you to color when we drew the plasmid, is LEC-Z gene. LEC-Z encodes for the enzyme beta-galactosidase, which will catalyze the hydrolysis of sugar. This sugar is lactose. Okay, benda yang sama kita belajar dalam 6.4 chapter sebelum ini adalah gene yang menghasilkan enzim yang akan hadam lactose. Okay, so remember these two important genes later. Now we're continuing with the second step. The second step is to cleave or cut the target DNA or plasmid. Both the target DNA and plasmid are cut by using the same restriction enzyme to produce compatible or complementary sticky end. Okay, kita akan potong kedua-dua target DNA dan plasmid menggunakan restriction enzyme yang sama supaya mereka mempunyai hujung yang berpatutan, yang compatible. Dia boleh melekat dengan satu sama lain. Okay, so you will use an enzyme that make a single cut within the LEC-Z gene of plasmid and many cuts within the target gene. So kita akan pilih restriction enzyme yang akan potong plasmid sekali sahaja, tapi yang akan potong target gene berkali-kali. Okay, kita akan potong target gene berkali-kali supaya dia akan ada segmen yang mengandungi benda yang kita mau saja. Plasmid kita potong sekali saja sebab cukup sekali untuk bagi dia buka dia punya ring itu. Therefore, the target DNA and the gene of interest and plasmid are now having complementary ends. Sebab kita potong dengan enzim yang betul, sekarang hujung dia boleh bersambung. Then we move on to the third step. Step number 3 is insertion. So insertion of gene of interest into plasmid. Since both the DNA of the plasmid and the target gene have compatible complementary sticky ends, they can pair with each other when mixed. So kita kasih campur dia. supaya the sticky end of gene of interest and cut plasmids are matched. Dia akan berjumpa. Bila dia berjumpa, dia adalah dimasukkan sudah. Okay, when it match, it is now inserted. And then dia belum terbentuk lagi phosphodiester bond di hujung ini. DNA ligase enzyme ataupun modifying enzyme akan datang untuk kasih sambung di sini, sambung di sini, sambung sini, dan sambung sini. Okay, DNA ligase will join these two fragments together, and the product formed is now called a recombinant DNA or recombinant plasmid. Selepas kita masukkan target gene, kita gam dia menggunakan DNA ligase, benda ini sekarang digelar sebagai recombinant DNA ataupun recombinant plasmid. Masa kita buat eksperimen tu, kita tidak akan nampak setiap plasmid yang terbentuk sebab kita tengok dengan mata kasar. So, masa kita campur-campur dalam test tube, ada yang berjaya insert dan menjadi recombinant plasmid. Tapi ada juga yang tidak berjaya dan mungkin dia akan jadi fragment DNA yang tidak berguna ataupun plasmid itu tidak dapat menerima target gene. Right? So this is another picture to illustrate how the insertion can occur. Ini adalah gambaran untuk menunjukkan macam mana insertion boleh berlaku. Sekali lagi, gambaran yang sama daripada isolation, cut or cleaving, and insertion. Okay, now we're going to move on into step number 4, which is transformation. Transformation is when we put that recombinant DNA into the host cell. The recombinant plasmid then is transferred or introduced into the bacterial cells through transformation process. Recombinant plasmid dimasukkan atau diperkenalkan kepada bacterial cell melalui proses transformation. Okay, transformation, now you don't need to learn about it in detail, tapi kalau kamu mau belajar maklumat tambahan, kadang-kadang transformation ini kita boleh menggunakan enzyme lain, kita boleh letakkan bahan-bahan lain ke dalam test tube, supaya itu host cell akan terima plasmid ke dalam. Kadang-kadang ada juga prosedur yang meminta kita kasih current itu test tube. So some procedures will require you to electrocute or to apply a little bit of electricity to the host cells so that they accept the plasmids. Tapi macam mana berlaku itu tidak termasuk dalam silibus kamu sekarang lah. Cukup setakat kamu tahu bila kita kasih masuk itu adalah transformation process. Okay, so after we transformed the host cells, we move on to step number 5. Step number 5 is amplification. Amplification is when the recombinant plasmid is amplified or multiplied in the host cell. So, mungkin host cell itu sudah cukup besar, E. coli itu sudah cukup besar, dia akan replicate, dia buat mitosis, dia buat binary fission, lepas itu dia jadi banyak. Bila dia menggandakan diri dia, plasmid pun akan digandakan. So this will result in multiple identical copies of the recombinant plasmids. And as the bacteria or host cell reproduce, they will form a colony and the recombinant plasmid will be cloned as well. Bila bakteria itu membiak untuk membentuk koloni, recombinant plasmid pun akan ikut digandakan, diklon. And after it forms colony, lepas kita biarkan bakteria itu untuk membiak, membesar, membanyak, kita perlu pilih mana yang berjaya terima target DNA dan mana yang gagal terima target DNA. Because like I said, some of the plasmids are successful and become recombinant plasmids, but some of them will be unsuccessful and may contain useless DNA. So only some of the bacteria may take up a plasmid. Not all the plasmids are recombinant plasmids. So some bacteria will take up plasmid while some will take up recombinant plasmid. The screening is important to identify clones of bacterial cells that contain recombinant plasmids only. Okay, kita mau cari bakteria yang mengandungi recombinant plasmid, yaitu plasmid yang kita sudah edit dan kita sudah masukkan gene of interest. To identify which host cells take up recombinant plasmids, all host cells are cultured or grown in a medium containing antibiotic ampicillin and sugar X-gal. Okay? Untuk mengenal pasti mana satu host cell yang berjaya menerima recombinant plasmid, semua bakteria yang kita dapat itu kita akan biak dia di dalam medium yang mengandungi antibiotik ampicillin dan gula yang bernama X-gal ataupun X-galactose. Okay, so there are 3 possible bacteria. One is non-transformed bacteria, the one without plasmid. Second is with plasmid but non-recombinant plasmid. The third is the transformed bacteria that contains the recombinant plasmid that we want. So this is a diagram that briefly illustrates how the blue-white screening is done. Pemilihan biru putih akan dilangsungkan seperti dalam diagram ini. Bila kita kasih tumbuh dia, kita biarkan dia membiak di dalam petri dish, dia ada kemungkinan untuk kelihatan seperti macam ni, ada warna biru, ada kemungkinan dia macam ini, langsung tidak apa-apa membiak, dan ada kemungkinan dia macam ini. Dia nampak warna putih dia punya koloni. Alright, so let's learn a bit more about this blue-white screening. How we do the blue-white screening is, you have your bacteria growing in a liquid solution. Okay, masa kita biarkan bacteria itu, kita kasih biar dia dalam liquid solution. Kemudian, kita akan plate dia kepada petri dish. So, you can have a petri dish. The petri dish will contain nutrients and also contain the antibiotic. Nutrient dia dalam bentuk X-galactosides, X-galactose, dan juga mengandungi antibiotik. So, kita ambil bakteria daripada cecair yang kita kasih biak tadi, kita kasih plate dia. Kasih letak saja di atas tu petri dish, dan biarkan petri dish itu selama 1 minggu, 2 minggu, sehingga bakteria membesar. So, that's what the first sentence means. You plate out the transformed bacteria into that nutrient medium. Antibiotic ampicillin is used to identify bacteria that have taken up plasmid. Kalau dia ada terima plasmid, sama ada plasmid yang kosong ataupun plasmid yang recombinant, dia boleh hidup dalam antibiotic. Only bacterial cells that have taken up a plasmid will survive and form colonies due to the antibiotic resistant gene, AMPR. Okay? So, kalau dia tidak terima sebarang plasmid langsung, dia tidak akan hidup. Kalau dia ada terima non-recombinant plasmid, dia hidup. Dia terima recombinant plasmid pun, dia akan hidup dalam medium tersebut. So, kalau dua-dua jenis ini hidup, macam mana kita tahu mana satu yang mengandungi recombinant, mana satu yang mengandungi plasmid biasa? Then we will go into the next type of screening, which is identifying which of them can digest X-gal. X-gal or X-galactose is a type of sugar that is used to identify bacteria that have taken up recombinant plasmid. X-gal bentuk dia sama dengan lactose, cuma berbeza sedikit. Okay? Now let's see how we use X-gal. Bacteria with no non-recombinant plasmids will have a functional LacZ gene due to no insertion of gene of interest. Kalau tiada gene of interest dimasukkan, that means LacZ gene dia tidak terkacau dan masih berfungsi seperti biasa. So the enzyme beta-galactosidase will be produced. Beta-galactosidase enzyme is able to metabolize or to digest X-gal sugar. Kalau dia dapat hadam X-gal sugar ini, X-gal sugar itu akan menyebabkan koloninya berwarna biru. If they can digest the X-gal sugar, the bacteria will form blue colonies on the plate. So if no gene of interest, Lexet functional, enzyme produced, able to metabolize X-gal sugar, form blue colonies. If your bacteria did not take up the normal plasmid but instead took up the recombinant plasmid, okay? Kalau bakteria kamu terdapat recombinant plasmid yang ada gene of interest di dalam, it will have non-functional Lexet gene due to insertion of gene of interest at multiple cloning site. Kalau ada gene of interest, dia akan kacau Lexet gene. Lexet gene kamu tidak berfungsi lagi. So the enzyme beta-galactosidase cannot be produced by this bacteria. X-gal sugar cannot be metabolized or broken down. Dia tidak akan dapat hadam X-gal sugar. And so it will form white colonies on the plate. Kalau X-gal tidak dihadam, bacterial colony akan membentuk koloni berwarna putih. So based on this, you know that in your Petri dish, you can have blue colored colony and white colored colony. Which color colony do you want to check? That's right. You want to take the white color colony because the white color colony will contain the gene of interest that you work so hard to insert. So what we have here is a table that briefly summarizes what happens in blue-white screening. So table ini, kalau kamu masih bingung, kamu boleh rujuk table ini. Row ini, yang without insertion ini, adalah sekiranya plasmid kamu tidak berjaya menerima target DNA. Macam mana dia punya target DNA? Tiada termasuk. Okay, Lexet gene dia berfungsi ke tidak? And then blah 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 blah. Final color dia blue. And the bottom row is what happens if your plasmid is able to receive the target gene. Kalau dia berjaya menerima target gene, macam mana dia punya Lexet gene, macam mana enzimnya, dan hasilnya adalah white colored colony. Alright, so once again, this is a diagram that illustrates how the blue-white screening functions and works. Okay, everything that I've taught you is referenced from chemical biology textbook. Alright, so that's it. Macam panjang lebar saya explain, tapi actually sangat ringkas. Ada 6 step sahaja dan asalkan kamu mampu untuk explain setiap 6 step itu, kamu okay. Alright, so just a very quick summary of the 6 steps involved in gene cloning. The first step is isolation of target DNA and plasmid or vector. The second step is to cut or cleave the target DNA and the vector using the same restriction enzyme. Number 3 is insertion of target DNA into the vector, and modifying enzyme will ensure that they are glued together, they stick together. Followed by transformation, where host cell is transformed by the recombinant DNA. Amplification, where the recombinant DNA together with the plasmid is amplified. And last but not least, screening. In this case, we learn about blue-white screening in order to choose the successful host cell. Okay, so remember the steps ichitas. And if you have any trouble with the steps or specifically with the screening process, feel free to ask a friend or ask me. So that is the end of 8.2. What should you do now? Now you should take the information from this lesson to create your own mind map of flowchart. Include any information that you want to remember well. Okay, apa yang kamu mau ingat, tolonglah masukkan dalam kamu punya mind map ataupun carta alir yang kamu mau gunakan. After you're done making your notes in the form of a mind map, complete tutorial question chapter 8, part C, question A. Buat soalan essay ini. So that is it for this video. Thank you very much for your time and attention, and I will see you again in the next lesson. Thank you. Bye!